Kia ora koutou, ko Douglas Walker, toko ingoa. Running through level 1 science genetics today. I'm very glad to have beside me Andrew Sargent, who you may have seen doing some of the physics webinars. And Andrew will take any questions that you've got and collate them. He might interrupt me or he might save questions till the end. So please do feel free to add questions into chat as we go. I would like to say a big thank you to Study It and the Ministry of Education. And I've put a little link in the description, and Andrew's put it in the chat. There's a survey over at Study It, which is a great website to go to if you need any help or want any questions answered for your level 1. And if you could do that for us, it would only take 2 minutes, and that would be much appreciated. All right, so we're here today for level 1 genetics. And to start us off with, I've mentioned Study It, and I'll make these slides available in the description after the webinar. There's also a really great site, I'm sure most of you know, which is No Brain Too Small. And that's got a huge number of resources, really good questions by type, along with the schedules and a whole heap of other things. I'll mention a couple of those as we go through. Okay, so things we're going to cover today. We're going to start off by looking at the mistakes that people have made in the past so that we don't make those same mistakes. I'll go through some practice exam questions from the 2020 paper, and I'll give you some advice for common approaches to how to best answer those questions. And with genetics, level 1 genetics, definitions are really important. And so you need to get to know a few key words that you are going to use throughout the exam. And so I've got linked in a page from No Brain Too Small. And so there's a great genetics glossary over there. You can go and check that out with all the keywords and definitions. And they've also made up some flashcards, which if you've got the time, I'd highly recommend printing out and use those. So have the definitions on one side, the word on the other, and test yourself or get somebody in your family to test you. They read out the key terms. You have to give the definition, for example. And knowing the key words in genetics is probably going to get you at least to an achieved level, and it's going to be very important for getting a merit or excellence grade. We'll cover meiosis in a reasonable bit of detail, and I'll touch on mitosis as well. We'll go through mutations, genotypes, and phenotypes, some key terms there. We'll go through an example of Punnett squares. I'd be very surprised if you didn't get a Punnett square in your exam. Pedigree charts, it's another one that's always in the level 1 genetics exam. We'll talk about variation. There's often a question relating to variation and natural selection. And we'll go over some key points about reproduction as well, particularly with reference to that variation and what that means for the survival of a population.
Just a question, Doug. Someone said, will there be one of these for microorganisms?
No, good question. There won't be one for microorganisms, I'm afraid. We haven't had any teachers in the area that are teaching it that have put their hand up to run one for microorganisms. So not this year, but I'd be hopeful that in future we'd be able to offer that. So learning from the mistakes of others. There's a report that comes out at the end of each year about the exam from the year before, and that contains a rundown from the examiners of the common mistakes that people make. It also covers what people do for achieved merit and excellence. I'm not going to worry about that because we're going to go through that in the questions that we work on today. What I am going to go through is the things that prevent people from achieving in the exam. So learn from others' mistakes and you'll do well. It's going to be quite a long list, so bear with me, but you can always go back and revisit these as you need to. So in the past, if students who got not achieved, they commonly didn't have a firm grasp of genetic language and confused some of the terms like homologous, homozygous, genotype and phenotype, or couldn't use terms like species variation, adaptation, gene, allele, and meiosis. We'll go through all of those today, but those are some key things that you want to make sure you're on top of. Again, terminology, heterozygous, homozygous, and I've thrown in there what the sort of an example of the genotypes might be for each of those. Quite an interesting misconception here. So students describe fertilization that occurred before or during meiosis. So just to be clear here, meiosis is the process by which gametes or sex cells are created. So that's sperm or eggs in humans. And they have to be created before fertilization can take place. So it's interesting to see that, a lot of students struggle with that idea and maybe confuse the two concepts. Students thought that variation was a response to something that happened in the environment rather than being caused by mixing of alleles through sexual reproduction. So variation is not in response to things happening in your environment. Variation is something that happens as part of sexual reproduction, and then that might give members of a population an advantage that then means that they've got a better chance of surviving certain environmental changes. In the past, students have demonstrated poor understanding of the causes and benefits of genetic variation within a population. So we'll look today at what things cause genetic variation and how that can benefit a population. Punnett squares, it's a key thing. So you've got to be able to complete the Punnett square and make sure that your Punnett square relates to what you're being asked in the question. There's a misconception, again, that students often perceive asexual reproduction as something that only happens in plants, or that plants only reproduce asexually. That's not the case. A lot of plants, especially flowering plants, are going to reproduce sexually. Some plants will reproduce both ways. So be aware of that and make sure that you know that that sexual reproduction can lead to variation within a population. Being able to use the resource material effectively. So that means being able to read and understand the question. When you're answering, don't just repeat what was in the question. You can use that to help you start your answer, but you really want to make sure that you have carefully read the question. And a pedigree chart, we'll go through these. You've got to be able to interpret and understand the pedigree chart. So I've put a link in there for where I got those from, and the concepts about genetics is a cool article on Science Learning Hub where you can go and read a little bit more about some of those common misconceptions. Might be a useful read before the exam. Okay, so that takes us to this image, and I've included this because it's a really good way of linking some of the key terms with a cell, nucleus, and so on and so forth. So I'll talk you through each of these. And this is just really key stuff about knowing those terms. You often get a question that asks you to relate these terms to each other. And you can show a lot of understanding simply by defining each term. But for higher levels, you want to link those terms together. So we've got the cell. That's the basic unit of life. We've got here an animal cell, and it contains a nucleus. And inside the nucleus is all the DNA. Most of the genetic information is found within the nucleus. And that's our chromosomes, so the genetic material on a chemical called deoxyribonucleic acid, DNA, and that the structures of DNA that are made of DNA are the chromosomes. So we've got 46 in our body cells, 23 in our gametes, our sex cells, as humans. We wouldn't normally see those as X's. It's normally only before meiosis starts that we would see those form into distinctive shapes. Normally, they're a kind of big mess of genetic material. And along the chromosome, it's made of a chemical called DNA. Different regions on the chromosome are genes. And one gene will provide the code to make a specific protein, which at level 1, we can say will normally code for a particular trait or characteristic. So the different regions of the chromosome, and there might be thousands on one chromosome. Each of those regions represents a gene. They can be differing lengths, and each gene codes for a specific protein. And the language that is used in DNA is the nucleotide bases. So that's your A's, T's, G's, and C's: adenine, thymine, guanine, cytosine. And it's important that you know how those pair up. So we've got adenine always pairs with thymine, cytosine with guanine. And if you want to just take a moment to have a think about the diagram that's there. So have a think about what the sequence would be in the blank grey boxes down the right-hand side. And the shapes there, the green circles and the pentagons, the orange pentagons, that's the sugar phosphate backbone of the DNA. And then the grey boxes represent the nucleotide bases that are normally joined through weak bonds in the center. So hopefully you figured out the correct order down there should be from the top, T, C, A, and G, and that's normally an achieved level point. So on to mitosis and meiosis. It's important to know the differences between them and to know a bit more detail, particularly about meiosis. So mitosis is the form of reproduction of cells, the duplication of cells, that's normally important in growth of tissue and replacing dead cells. So mitosis has one division, so it's where one cell duplicates into two genetically identical daughter cells. And that produces our body cells. And if you're trying to remember which one's which, because they conveniently both start with M, then mitosis happens in your toes. So mitosis, my toes, your toes are part of your body cells, and so that's happening in your non-sex cells. We also talk about those body cells as being somatic cells, so you can use that term as well. Meiosis is a bit more complex. So what happens there is there are two stages of division, and the end product will be 4 haploid sex cells or gametes. Haploid meaning that is half of the genetic number. So if we as humans start with 46 at the top of meiosis there, then in the bottom here, these 4 cells would each have 23. So 46 up here, and each of these would have 23. So that's what we mean by haploid. It's half of the normal genetic number. Comparing that to mitosis, we talk about these cells being diploid, which is they've got the full genetic component. So in mitosis, if we started with 46, both of these cells are genetically identical and have 46 chromosomes each in humans. More to say about that. It's the 4 haploid cells, the sex cells that are produced, the sperm or the eggs, or the pollen or ova in plants perhaps. They're genetically unique, and so every single one will be different, which is quite amazing, especially considering that in the average male ejaculation in humans, you're producing about 300 to 400 sperm, and each of those is genetically unique. It's quite unlikely throughout the course of a lifetime that a human male would produce one sperm that is the same as any other that he's produced in his lifetime. So meiosis has 2 stages of division, and we have a couple of extra features in there, namely independent assortment, segregation, and crossing over. Now, hopefully you're familiar with those terms, but I'll talk about them in more detail. And just a note there that crossing over, your teachers might have talked about that as being recombination. Now, this is quite a complex looking diagram, so hopefully you won't be put off, but I put it in here because it's probably the best one I could find that didn't have any little errors in it. So we start off with the diploid cell, and then arrow comes through here. We're here. This is where we're lining the homologous chromosomes are pairing up. So that word homologous, we can generally take it to mean chromosomes of the same length. And so you'll have 2 chromosomes in each of most of your cells that will be the same length, and they will pair up as homologous chromosomes. To give you a little bit more detail about that, the homologous actually means that the genes that are found on one of those chromosomes normally have matching or corresponding genes on the other chromosome in similar regions. So if you imagined at the very end of the very tip of this purple chromosome, then there would be matching genes on the tip of the blue chromosome, maybe for a protein that contributes towards eye color. So homologous chromosomes pair up, and then we've got this stage here where they're lined up along the equator of the cell. We've got the little centromeres, these are the gray dots out to the side, and in a moment they're going to do something quite important. But for the moment, we've just got those homologous pairs of chromosomes lining up down what we call the equator of the cell. And so they're lining up in pairs of similar length all the way down the equator of the cell. Now, the independent assortment means that they can line up in different orientations. So it might be that in one instance, the purple chromosome is on the left and the blue is on the right. And for the next pair, the blue is on the left, but the purple is on the right. Now, we're dealing with 23 pairs of chromosomes. So as you go down there, it's effectively the independent assortment is the random chance that each of those purple chromosomes, if you like, will be on the left or the right. And that has an important role to play in how variation comes about through meiosis. So that's independent assortment. Then we've got this stage here where the chromosomes are being pulled apart towards the poles of the cell. So if the equator in this diagram was through the middle here, then we would identify these bits as being the poles of the cell. And the poles pull those chromosomes apart, normally with one chromosome going each way. And that is segregation. So it's the pulling apart. And that segregation is another way that variation can creep in. And it goes hand in hand with that independent assortment. If you didn't have independent assortment, then you'd always be pulling the same chromosomes to one side of the cell and the other chromosomes to the other side. So independent assortment and segregation work together to separate random mixtures of chromosomes. We then, the other thing I haven't talked about over here is crossing over. So when those homologous chromosomes are lined up on the equator, you can see that the legs are quite close together. And in some cases, they overlap and swap over. We'll have a look at that more closely in a moment. But for just now, you can hopefully see on the screen there that there is a little bit of blue and a little bit of purple, and they've swapped over. You might be able to see it more clearly down here, where the purple chromosome has taken a bit of the blue, and the blue chromosome has taken a bit of the purple. And so that means that genes that are on a particular chromosome can be swapped around with a neighboring chromosome, and that increases the variation that comes about through meiosis. Okay, and again, the end product, I'm not too worried about what's going on in here. That's just the second stage of division to produce 4 genetically unique haploid cells. And got some little pointers on there for independent assortment, crossing over, and segregation. It's useful to know a bit about each of those. I've put in this diagram just to highlight the different ways that independent assortment can produce different outcomes. So in possibility 1, we've got the blue lined up on the left, the red lined up on the right. It's obviously very simplified if it's a human example, because we've only got 2 pairs of chromosomes here, whereas we would have 23 pairs of chromosomes. But that's going to be a very confusing diagram if we did it like that. So in possibility 2, we can see that some of those chromosomes have swapped about, and that's what we're talking about with independent assortment. So we can have, one chromosome on the left, and then in another situation, that chromosome might be on the right, which means that when they're segregated, that's going to produce 2 different cells, and then they'll go through division again, which will produce different combinations of end product of sexual cells, gametes. And here's a bit of a look at crossing over. So just zooming in a bit more detail, so you can see that when those homologous chromosomes line up, then we get sorry, I just got a bell ringing in my ear here. When those homologous chromosomes line up down the equator of the cell, then the more often the end pieces, the legs, cross over and they actually swap over. And so we can see here that the blue chromosome has picked up a bit of the red chromosome has picked up a bit of the blue. And so whatever genes were in this blue part are now tied to this red chromosome, which is really important in terms of being able to produce as much variation as you can. In fact, I think it's about, if you didn't have crossing over, there'd only be about 8.5 million different combinations when you go through independent assortment and segregation. So crossing over really amplifies that. And if you think about what I said earlier about the number of sperm that are produced in an average ejaculation, then you're dealing with 3 to 400 million. And if there was only 8.5 million possible combinations, then a lot of those sperm would end up being identical. But crossing over adds to that level of variation. Interestingly, it happens more often in females. It happens about 75 times per cycle of meiosis in females, but only 55 times in males. Okay, so we're going to look at how some of those things fit together in an exam, and that's going to give us an opportunity to cover some of the other areas that we might need to look at. So I've got the 2020 paper here. Just pulled that out because we've got some other ones from older papers, and if you have a look, you'll be able to find videos on those. So going through, making sure that you're reading the question carefully. We've got a condition called Huntington's disease. We've got a dominant affected allele, it's the capital H, the dominant showing that it's shown by capital, and the normal allele is recessive, which is the lowercase h. We're asked to state the possible genotypes and phenotypes for Huntington's disease. And so that's really just asking us to put down all the possible combinations of genotype and what their phenotype means. So it's quite a low level, it's going to be an achieved question. And we should know that we can pair 2 capital H's together, 2 lowercase h's together, or we can take one of each. So we'll write those in down the side. Any genotype that has a dominant allele present will show that characteristic. And so here the phenotype for number 1 is going to be affected by Huntington's disease. The second genotype still has 1 dominant, doesn't matter that it's got single recessive, still going to express that affected by Huntington's disease. And then what we call the homozygous recessive, the 2 lowercase h's, means that there is no dominant allele that's going to create Huntington's disease or make the individual affected by Huntington's disease. And so that person is going to be what we describe as normal in this situation. So just some key terms there. The first genotype is homozygous dominant, 2 of the same allele, and they're both dominant. Number 2 is heterozygous, 1 of each, dominant and recessive. And number 3 is homozygous recessive. So 2 of the same, but they're the recessive or lowercase letters. And that's an achieved level question. Okay, so how do the 2 alleles combine to produce different phenotypes? So remembering that the phenotype is the way that a trait is expressed. Sometimes we often level 1 talk about it being visible, but it might be something that's happening inside the body that isn't so visible, but it's still going to cause an effect. So we need to define phenotype and genotype, and we need to explain how the 3 genotypes code for only 2 phenotypes. I've kind of covered that already verbally, but we'll go through it a little bit more carefully here. So first off, we'll do what the question's asked us. We're going to define phenotype. And after I've done that, if I was in the exam, I would tick that off on the paper just as a sort of way of checking for myself to make sure I've done what I've been asked. So phenotype is the expressed trait or characteristic based on the individual's alleles. The genotype is the combination of alleles for that particular trait. And so we express that as 2 letters. And then in terms of the explain, so now I've ticked off phenotype, I've ticked off genotype. Explain how the 3 genotypes code for only 2 phenotypes. Alleles are either dominant or recessive. A dominant allele will always be expressed if present in the genotype. So homozygous dominant or heterozygous will result in the same phenotype. In this case, that would be affected by Huntington's disease. However, a recessive allele is only going to be expressed if it is the only alleles that are in that genotype. If a dominant allele is absent, that means so if there's no dominant allele and there's only recessive alleles, then that allele will be expressed, that trait will be expressed. And so the genotype little h or homozygous recessive provides the unaffected phenotype. And that's an excellent question. I'd point out here that you don't actually have to write a whole heap. If you look at the paper, there's a lot of space for this. But if you have practiced your responses to common exam questions, I would do that by going back through the last 5 years' papers for genetics. Then you're going to see the same questions again and again, and you're going to be able to refine your answers. So you're doing what the question's asked, you're doing it in a reasonably concise way, so you don't end up waffling, and use your time wisely instead. All right, sorry if there's a bit of background noise coming through here. Huntington's disease occurs when a mutation causes a dominant affected allele. The normal allele is recessive. And quite a common question here that's leading up to telling us what a mutation is. So using Huntington's disease as an example, we've got to explain the relationship between DNA, genes, alleles, mutations, and phenotype. You can expect some sort of question that's going to ask you to tie together some of the key terms in genetics. Easy thing to do is to define each of those, and that's where your flashcards are going to come in really useful. And then for the higher grades, you've got to be able to link them together to show that you really understand what's going on there. Okay, so just for space, I've moved that question up to the top. All right, so a mutation, what is it? You should have this definition ready to go. It's a random permanent change in the series of bases of our DNA. A gene is a region of DNA that provides the code for one specific protein. Now, if there's a mutation, that can create a different version of a gene, a new version of the gene, if you like, a new allele. We normally, in level 1, only talk about 2 alleles for any particular trait, but in reality, in a population, you might have 20 different alleles for a particular characteristic, and they've all come about through mutation. Every allele that we possess, that any living thing possesses, has come about through mutation at some point in the past. And so a new allele could produce a different protein. In this case, that new allele does produce a different protein, and that different protein causes the condition known as Huntington's disease. And if that trait is expressed, then that is what we identify as the phenotype. So based on the combination of alleles that are inherited from the parents, that is the phenotype. And the question asked us to use the diagram above. So in our answer, we might refer back to we could identify on the diagram the particular regions here. So that region is a gene, and the 2 different versions of that gene are alleles. And the chemical that makes up these chromosomes is the DNA. Feel free to annotate the diagrams in the exam. It's a really good idea. And to draw your own diagrams if it helps. Now, importantly, and I don't think this is necessarily important for this question, but I wanted to throw it in there, a mutation must occur in the gamete or the cells that produce gametes in order for the mutation to be passed on to offspring. And then, if that's the case, that mutation will be present in each. So that new allele that's come about through mutation will be present in each of the offspring cells. And in that way, a new allele can enter a population. From the moment we're born, in fact, possibly even from before we're born, our cells can mutate. And so we accumulate mutations throughout the course of our lives. That's often what ultimately cancer is. And it's a natural part of life in that our cells sometimes make mistakes during copying, and those mutations creep in. It's quite rare for those mutations to happen inside our sex cells and then be passed on onto offspring. So, you might pick up a lot of mutations. Most of us, certainly by my age, I'm going to have lots of cells that have mutated in some way. Most of the time, that's not an issue at all. But they're not going to be passed on to my kids unless that was to happen in my gametes or in the cells that produce my gametes. And that's an excellent answer there. Okay, so pedigree charts. Put money on having a pedigree chart in the exam, I would say. And this one combines with a Punnett square, which it tends to be in the exam. really important that you carefully read the question and you make sure you understand the key and how that applies to the situation here. Lots of practice with this one. Go through old exam papers, have a look on No Brain Too Small, and check your answers as you go through to make sure you're understanding the concepts. Okay, so we're being asked to complete a Punnett square for the cross between individual 1, who we can see is from the square part, he's a male, and he is unaffected because it's a white box. Number 2, we're told is heterozygous. That means 1 dominant allele, 1 recessive allele for polycystic kidney disease. And so there's a little picture of polycystic kidney disease there. There's a lot of cysts that grow on the kidneys, and it becomes very problematic. So we know that number 2 is female due to the circle and affected because of the filled in black color there. Okay, so then we've got our Punnett square, and what we're going to do is we're going to write in our genotypes for number 1 and 2, and so you can tell from that the homozygous recessive is my number 1 individual, and then the heterozygous, as stated in the question, is my number 2 individual. The next job is to separate out those alleles into the smaller circles, and so we've got the dominant allele D, big D, and the recessive little d in the blue boxes, in the blue circles down there. And then we've got the 2 recessive alleles from parent number 1. And then we're going to fill out the Punnett square by putting in the letters in each of the boxes in the Punnett square. So what I would normally do is start off with one of the parents, and then I would write each of those, the letters from those in the corresponding row of my Punnett square. So let's have a look at what that should look like. I've got my dominant alleles from the big blue D, and then I've got the recessive alleles there. And then what's going to happen is we're going to pair those up with the alleles from the other parent. And so again, just fill in, should have really done that in columns, but hopefully you get the idea. And so that gives us an expected 50% of the population, not population, sorry, 50% of offspring would be expected to be heterozygous, or in this case, affected by polycystic kidney disease. And 50% of the offspring, 2 boxes out of 4, would be unaffected. How are we doing for questions, Andrew?
Good. No questions so far. Just students were asking about links to the other streams, so I just chucked all the links into the chat.
Awesome. Thank you for doing that. Okay, and this one, interestingly, was two achieved points in the paper, because you got 1 for doing the Punnett square, and then if you got the Punnett square right, your answer from the Punnett square could be taken forwards for part B as well. But normally I'd expect filling out Punnett square to be an achieved level, and then you probably have to expand on that in some way, which will be your merit and excellence opportunities. Okay, and so our next job is to list the genotypes of another 2 individuals, number 3 and 4. And so this is where a little bit of detective work comes in, interpreting the pedigree chart and understanding what's going on there. Individual 4 is pretty easy because they're unaffected. They can only be homozygous recessive, little d, little d. And just a point, use the letters they give you in the question. So quite often you get so used to using letters from other examples, you forget in the heat of the moment in the exam, but read the question carefully and use the letters they give you. All right, individual 3 is perhaps a little bit more problematic, but if we go back and have a look at our Punnett square, then we know that the offspring can only be either heterozygous or homozygous recessive. Individual 3 we can see is affected. It's a male who is affected by polycystic kidney disease. And so they must be heterozygous. That's the 1 of each. Okay, and that's an achieved level. So moving on to part C, and we see that keyword explain. That suggests that we're looking at a much higher level answer, a merit or an excellence opportunity coming along. Now, you can see in the question that they've put in capital letters, both and so I would highlight those and remind myself to come back to them, because when I've finished a question, I would go back and reread the question to make sure I've answered everything that has been asked of me. So how did we work out the genotype for individual 3? Well, we could do it with just the parents, but we've been asked to do it with the children as well. And so we've got to make sure that we do both of those things. So we know that the father's genotype was homozygous recessive, little d, and he can only pass on one allele to his offspring. And so number 3 must have got a recessive allele from dad. The mother's genotype, I think I put in here either homozygous dominant or heterozygous, and I think I just realized I might have made a mistake there, and I want to go back up to the start of the question. We're told that individual 2 is heterozygous, so you can see that down in the question here. And so actually, while it doesn't really change my answer, I'll just point out that that should say the mother's genotype can't be homozygous dominant. It can only be heterozygous. But because individual 3 is affected by the disease, then they must have got the dominant allele, and that must have come from individual number 2, the mother. So as 3 is affected, he must carry at least one dominant allele, and that could only have been inherited from mum because dad only had 2 of the recessive ones. Okay, so we've effectively there's no two ways about that. We've definitively explained the genotype of number 3. However, it did ask us to do the children as well, so we'll go ahead and do that too. So number 7, we know the genotype is homozygous recessive, which means that number 7 must have received the recessive allele from both parents 3 and 4. It's very clear how it came from 4, but it tells us that number 3 must be heterozygous to have been able to pass on one recessive allele. And then 8 and 9 are both affected, so they must have inherited a dominant allele from one of their parents. Can't have come from number 4, can only have come from their father, number 3 in this case. And so that again reinforces that number 3 must be heterozygous. Okay, again, go back, reread the question, check that you've done everything that's expected of you, and that is an excellent response. So one of the family members has kidney failure as a result of an infection. Discuss if this type of kidney failure can be inherited by any future children they have. So I talked earlier about how a mutation has to occur in the gametes or in the cells that produce gametes in order for it to be passed on to offspring. That's not the case with an infection. So the infection is very much only affecting the somatic cells of the individual, and so can't be passed on to the offspring genetically. And this brings in the idea of environmental versus inherited characteristics, if you like. So environmental, the infection is a result of the environment, it's not being inherited. So not caused by the genes of the infected individual. And because it's not caused by the genes, then it's not going to be passed on to offspring. So genetic information is only passed on to offspring through gametes. And as the kidney infection affects somatic, the body cells, and is not caused by the individual's alleles, it cannot be inherited by future children. And that is an excellence level response. Again, have a look at how much you've actually got to say and try and keep your answers reasonably concise so you maximize the time that you've got in the exam to reread questions, to reread your own answers, make sure you've added all the detail you can. And of course, if you're doing the other papers, that gives you time to do the same for the other papers as well. All right, so question 3. Malaria is quite fascinating, and I was half tempted to write a little question for you on human malaria and sickle cell anemia, but we'll stick to the one that was in the exam. So avian malaria, avian just being to do with birds, is a parasitic disease affecting hoiho, so the yellow-eyed penguins, and that can lead to their death. So there's 3 parts to this question. It's describe genetic variation, and then we've got an explain question, but we'll come on and have a look at that in a bit more detail after we've done A. So describe is a fairly low level response. It's normally an achieved level response. And so we're only being asked to describe genetic variation. And it took me a moment actually to think, what's the question actually asking me to do here? And it's really just saying, look, tell us how hoiho are different. So it's the differences in the traits, whether visible or hidden, of the penguin that result from combinations of alleles on their chromosomes. And I probably didn't need the last part of that sentence, but thrown it in there anyway. So genetic variation is the differences in traits, visible or hidden, that result from combinations of alleles on their chromosomes. It's an achieved level response. And then part B, I've highlighted the explain there. Again, good strategy to highlight keywords in your questions so that you think about what sort of level you've got to go into. And the space in the exam paper also gives you a clue as to how much is expected of you. Explain how sexual reproduction causes genetic variation in the hoi-ho population. In your answer, you should consider the process of gamete formation, meiosis, and fertilization. You may use labeled diagrams with notes to support your answer. And so you could go back at this stage if you wanted to recap that before having to go at the answer yourself. It would be a really good idea. You could go back up to the diagram I had that showed the differences between mitosis and meiosis. And a reminder here that in the assessment report, we were told that a lot of people get confused about when meiosis happens and when fertilization happens and where the contribution to variation creeps in. So a reminder that meiosis has to happen before fertilization because meiosis produces sex cells, and it's those sex cells that then go on to perform fertilization to create the zygote, the cell that's formed after fertilization, which becomes a new organism. All right. So sexual reproduction, it's creating a new organism through random fertilization between a male and female gamete. During meiosis, those three things we talked about earlier, the independent assortment, segregation, and crossing over, all contribute to create a genetically unique haploid gamete. And we make a lot of those haploid gametes. Female humans start with about 400 when they're born, and so you've got those throughout, from the day you're born, but they mature and aren't released until later in your life. Males are producing gametes constantly from puberty throughout the rest of their lives. Each of the gametes carries a different mixture of the parental alleles, the alleles that came from the parents. Each gamete contains half the genetic information. Remember, that's that word haploid. And these recombine in the zygote to give a mixture of alleles that will determine the traits of the offspring. Okay, I haven't included the diagram here because it's included elsewhere. And then C is asking us to discuss. Discuss is quite high level. I'd be looking at excellence for that. Part B is excellence as well. And here's our part C. So genetic variation improves, sorry, I should read the question. Discuss how genetic variation could lead to increased survival of the population when faced with avian malaria. Okay, again, classic question. I would be very well prepared for this going into the exam because there's a good chance it's going to come at you in some form. So having genetic variation improves the chance for some members of a population to survive when things change in their environment. Now, that might be a disease in the environment, or it might be a change in temperature. It might be a shortage of a particular food supply. It could be drought, could be all sorts of different things. In comparison, if a population was all genetically identical, if every member was particularly susceptible to a disease, then that disease could rip through the population and completely obliterate, if you like, kill all members of the population. Similarly, if we look at crop plants throughout the world, then some of those crop plants are genetically identical because farmers want particular traits or characteristics that are favorable. But if the conditions change, then that genetically identical population could become very susceptible to any change in the environmental conditions. So generally, variation is going to be an advantage because it means that some members of the population can survive and go on to pass on their successful genes, and then members of the subsequent generations will carry more of those alleles that have made that previous individual successful. So those with the alleles that are better suited to the new environmental conditions in this case avian malaria, will be more likely to survive and reproduce. That's a key part that people often miss out in their answer. So it's that ability to not only survive, but to reproduce and pass on your alleles or the successful alleles to subsequent generations. Sudden changes in the environmental conditions can be detrimental, bad, and in some cases lethal, but variation improves the chances that some members of a population will survive. Over generations, many, many generations, more individuals in the population will have beneficial alleles, leading to increased survival of that population. And if you enjoy watching biology-related videos, I can strongly recommend Galapagos. It's a 3-part series by David Attenborough, and that covers this concept really elegantly. And it's a very well filmed, very well produced documentary that you probably enjoy anyway. All right, now I think that brings us to the end. That's our excellence of the material that I've prepared for you. Here's a quick look back at what we've covered just before we have time for any other questions. So we learned from the mistakes of others. You can go back and have a look at that document about the common mistakes that cause people to get not achieved. I haven't covered the achieved, merit, and excellence in that slide because we've covered those criteria as we've gone through example questions and concepts. I've tried to cover some advice for common approaches to answering questions. The one thing that I maybe didn't mention was that it's useful to jot down sometimes, especially for those longer answers, the keywords that you're going to use in your answer, and then you can tick those off as you've gone through and used them. And whenever you use a keyword, it's a good idea to define it. And coming to definitions, we talked about those earlier, but you want to brush up on those, particularly for genetics. We've covered meiosis and mitosis. You don't need to know a lot of detail about mitosis, but it's important to know the differences between mitosis and meiosis. You need to know a little bit more about what goes on in meiosis so that you can talk about where variation creeps in to sexual reproduction and to the formation of those gametes. We've talked about mutations, get a definition ready to go for a mutation. Genotypes and phenotypes, you've got to know what those are and how to read, interpret, and use them. Punnett squares, hugely important. Pedigree charts, I'd expect to see it in the exam. Variation, an important concept that normally comes up in the exam. And reproduction, we've talked about throughout in a number of different areas.
Someone just asked if you could explain variation again.
Yeah, absolutely. So I'll go back up to here. So variation, genetic variation is the differences in combinations of alleles. So that's the letters that we normally write for the genotypes. We could also talk about variation in the phenotype, and the phenotype is normally the expression of the genotype. So at level 1 science, it's what we often think about as what you see. So it might be the shape of the nose, the shape of the ears, the color of the hair, the eyes. You know, those are the things that are your phenotype. And so you can see variation there. And so phenotypic variation is the differences in characteristics. Genetic variation is the differences in the genes that an individual has. And that might be one individual within a population that has, you know, different members of the population have a lot of different genes. And so there might be huge genetic variation in a population, or in some cases, you might have small populations that are quite closely related, and then you don't have as much genetic variation. I'm not sure if I've answered the question. Would you like to clarify that question any further or ask anything else about variation that I could drill into a bit more depth for you? You might have done an experiment in class at the start of genetics where you measured different characteristics of people in the class. And that's a really classic introduction to variation. So looking at all of the differences in the people around you and, you know, maybe measuring those. So it might have been, what sort of things would we do? It might be foot length. It might be whether, you know, how far back you can bend your thumb. It might be whether you can roll your tongue, eye color, etc. So all of those differences is what we call variation. And we can talk
someone
level
asked
level.
Someone asked if you could explain pedigree charts more, and the person who asked about the variation said their explanation was fine.
Okay, so pedigree charts. I'm not sure that I'll be able to pull up a different question on the hoof, but if I come back to this one, they're all very, very similar. So coming back up here, looking at the pedigree chart, what more could I say about that? Well, the numbers are normally there to help you refer to individuals in your answer. So you don't need to say the one at the top left. You can just refer to the number. And each of the shapes tells you something about, in this case, the gender of the individual. So the square is male. We can see that in the key here. And the circle is female. And then the way that they're shaded normally tells you about whether they express a phenotype or whether they do not express that phenotype. We would then look at the individuals on the pedigree chart, and we would have to make some, we would have to decide using evidence in the chart, perhaps to work out the particular genotypes of each individual. We know the phenotypes because we're being told them, whether they're affected or whether they're unaffected in this case, but we don't know the genotypes, and that's the skill that you'll often be asked to demonstrate. So if I looked at number 6, for example, we know that number 6 is a child of the parents 1 and 2. And so looking at that, number 6, I could figure out very quickly what their genotype is because I know that dad was homozygous recessive. He's unaffected, which means he must be carrying little d, little d. He can only pass on one of those alleles, and they're both the same. So any offspring that number 1 has is going to carry a little d. So we know that number 6 has a lowercase d, the recessive allele. We know that number 6 is affected, and they can only be affected because the kidney disease is caused by a dominant allele. They must possess at least one copy of the dominant allele. And so they must be big D, little d, or heterozygous as their genotype. And the dominant allele could only have come from their mum in this case. Number 5 must be the same. Number 5 is another offspring of parents 1 and 2. And he, because we know he's a male, is affected. And he also inherited the dominant allele from number 2. He inherited the recessive allele from dad because dad can't give him any other allele. Number 4 has come in from somewhere else. Okay, so number 3 is the third child or offspring of parents 1 and 2. I say third, 1 of 3. But number 4 is somebody who's from a different family group. Okay, but again, looking at the image there, we know it's a circle, so it's a female, and we know they're unaffected because it's not shaded in, and so their genotype must be homozygous recessive, little d, little d. And I think now I've kind of gone through all of the genotypes of the pedigree chart there, either in the question earlier or in response there. Does that give you what you need, or do you want to ask me a more specific question? We've got plenty of time for questions if you do have any others.
I said I covered the question fine.
Cool. Thank you. Good question. And it's an important skill to master for your exam. As I've said before, no brain too small. It's got questions organized on pedigree charts, so you can go and practice one, and then once you've done it to the best of your ability, maybe use your notes and flesh out any details that you might have missed. Then check the schedule, and then look at what you've learned. I would even do it in different colors. I'd do first time what I already knew in one color. Then when I use my notes, I might use a different colored pen just to highlight how much I know, how much I've relied on my notes for. And then when I check the schedule, I'd add any additional information. Once you've done that, you're going to improve what you understand about that question. And then you can go on and do another practice one. And again, what you know, what you've had to add from your notes, and then anything that's come from finally looking at the schedule. And as you do 3 or 4 of those, you'll see that over time, you'll be able to do more and more yourself without having to refer to your notes. And you'll see the patterns that come up. You know, there's only so many questions they can ask you in genetics. So practice really does make perfect.
Someone asked the question, what points should you cover for how sexual reproduction leads to variation?
How sexual reproduction leads to variation. Okay, so it's saying sexual reproduction. So I probably wouldn't go into mutation in too much detail in that question, but it's important to note that mutation is fundamentally the real introducer of variation. However, if the question specifically stating sexual reproduction, then it's probably looking for independent assortment, segregation, and crossing over. Those are the 3 things that contribute towards genetic variation as part of sexual reproduction. The mutation's already got to have happened somewhere in order for that to be affected by those 3 things. And so I'll go back up to all right, those were the 3 key steps. Independent assortment, again, is where the homologous chromosomes, so the same size chromosomes, they line up on the equator, but whether the one that you got from dad is on the left-hand side or whether you got the one from mum is on the left-hand side is what we're talking about independent assortment. So it's like a randomization process of all of your chromosomes. And because you've got 23 pairs of chromosomes, if you crunch the numbers, it comes out about 8.5 million different possible combinations. So if you on your calculator, you could do 2 to the power of 23, and that gives you the number of different possible combinations as a result of independent assortment. Segregation then pulls them apart to different ends of the cell. And so that separating them out means that different chromosomes will be used to produce the alleles that are in a particular gamete, a particular sex cell. And then finally crossing over. Crossing over is going to happen between those 2 stages. You don't really need to know too much about, when they occur during meiosis. It's more about that they occur and that they contribute towards variation. And crossing over was that one where when the homologous chromosomes are lined up, tends to be more towards the ends of the chromosomes, those loose ends. They cross over and they swap over their genetic material, which means that you're not just passing on either dad's chromosome or mum's chromosome, you're actually passing on a new chromosome that has mixed parts of mum's chromosome with parts of dad's chromosome. Does that help?
They asked, do you not need to mention the random event of fertilization between the 2 unique gametes?
That's a really good call. Yeah, I missed that one out. And so, as I mentioned earlier, you've got, male ejaculation's got 300 to 400 million sperm, and you get one winner from that race. And so it's going to be a random event as to which of those sperm fertilizes the egg. Obviously, all of those processes that we've already talked about have determined which alleles that particular sperm is carrying. So yes, it would be good to refer to that random event, that fertilization as a random event. Well done on picking that up. Any other questions? Remember that there's that website, Study It. If you've got specific questions, you can jump on there, and there'll be teachers across New Zealand who'll be able to answer your questions for any of your, I think there's science, English, and maths on there. And that's something you might use in future years as well. All right, if there's no other questions, I think we've hit 3 o'clock. It's been lovely talking to you. Thank you very much for your questions. Best wishes for your exam. And we've got I think it's acids and bases at 4 o'clock tomorrow, and the mechanics at 5:10 tomorrow as well. And the recording of this you'll be able to access with the same link that you've used to access the webinar. All right.
Someone just quickly asked, could you please explain fertilization? don't know if we've got time for that.
Yeah, absolutely. Let's make time for it, shall we? So fertilization. So a male is going to produce sperm. A female is going to produce an egg. The egg is the largest cell in the human body. It's the size of a dot. So if you take your pen and you put a dot on a paper, that's about the size of the egg cell. Fertilization is where the sperm has swam through the urethra. Through the urethra. Oh, whoops! No, not through the urethra. The sperm has entered through the vagina. It's then travelled up through the womb, through the oviduct or the fallopian tube, and then that sperm, in a big race with all the other sperm, is going to meet the egg. It's got probably too much detail for you here, but it's got a little enzyme in the tip of its head, and that enzyme is used to break through the outside of the egg, and then the sperm inserts that little bit of haploid DNA, so the nucleus, which is only half the genetic component, 23 chromosomes in humans. It's going to insert that into the egg, and then you've got the 23 chromosomes from the mother and the 23 chromosomes in the nucleus from the sperm. They're both now inside the egg. They join and fuse together, and that process of fusing together is called fertilization. You've then got one egg cell, and that egg cell now has the diploid number. It's got 46 pairs of chromosomes, and it's going to then go on to divide and divide and divide and grow, and that's going to become a genetically unique individual. Does that answer your question? Okay.
Yeah, they said yes.
Awesome. All right. Well, all the best, guys, and wishing you well for your revision. Do the hard yards now, and I'm sure you'll feel better about it when you're doing your exam. Great having you along. Ka kite. Thank you, Andrew.